I am Dr. C. Swaminathan, Assistant Professor of Microbiology, St.Joseph College of Arts and Science, Cuddalore. In this video, we are going to discuss about bacterial transformation. Let us see the definition for bacterial transformation. Transformation is a process in which DNA from the exogenous source is taken in and incorporated into the genome of a recipient bacterial cell. Transformation process was discovered by Frederick Griffith in the year 1928. Competence in transformation. Competence is a specialized physiological state of the bacteria in which they can take up the DNA from the environment. Transformation frequency of competent cell is 10⁻³ for most of the genera. I mean, one cell in every 1,000 will take up the DNA and integrate the gene. Competency is a complex phenomenon. It depends upon certain conditions. Competency usually arises at a late log phase or stationary phase. Competency is also triggered by nutritional shortages or oxidative stress. Types of competence. There are two types of competence: natural competence and artificial competence. First, we are going to discuss about the natural competence. Naturally competent bacterium are able to take up the DNA from the environment. They won't require any special treatment. Natural transformation has been discovered in certain genera only. For example, Streptococcus, Bacillus, Thermoactinomyces, Haemophilus, Neisseria, Moraxella, Acinetobacter, Acetobacter, Helicobacter, and Pseudomonas. Bacterial cells which are in starving condition requires nucleotides for their own DNA and RNA synthesis. So they become naturally competent and take up the DNA molecule from the environment and utilize the nucleotides for their own synthesis. The exogenous DNA molecules are also used for the repair of damaged DNA molecules. Next one is the artificial competence. Artificial competence is induced in the laboratory by means of exposing the bacterium to the conditions that would not normally occur in nature. Natural transformation. Gram-positive bacteria and gram-negative bacteria differs in the structure of their envelope. So thus there are certain differences in the mechanism of DNA uptake in the cell. Now we are going to see the basic steps which are involved in transformation. First step is binding of double-stranded DNA molecule to the outer cell surface of the bacterium. Second one is movement of DNA across the cell wall and outer membrane. Third step is degradation of one of the DNA strands. Fourth step is translocation of the remaining single strand of DNA into the cytoplasm of the cell across the inner membrane. The final step is the single-stranded DNA molecule might synthesize the complementary strand and re-establish itself as a plasmid, stably integrate into the chromosome, or may get degraded. Bacterial transformation as seen in Streptococcus pneumoniae. The mechanism of transformation has been extensively studied in Streptococcus pneumoniae. Competent cell has lac minus gene in the bacterial chromosome. Donor DNA fragment with a lac plus gene binds to the cell surface receptor. Extracellular endonuclease enzyme cuts the DNA into small fragments. One strand of the DNA is degraded and a single strand is transported into the cell through the uptake system. Single-stranded DNA molecule aligns itself with a homologous region on the bacterial chromosome. The single-stranded DNA is then incorporated into the bacterial chromosome via homologous recombination. The heteroduplex DNA is then repaired in a way that changes the lac minus strand to create a lac plus gene. Now we are going to discuss about DNA uptake machinery in Neisseria gonorrhoeae. Neisseria gonorrhoeae is a gram-negative bacterium. Pile Q aids in the movement of DNA molecule across the outer membrane. Pile E is the pyline complex that moves the DNA molecule through the periplasm and peptidoglycan. Double-stranded DNA molecule binds to COM-E. COM-E is the DNA binding protein. Nucleus, that is the N, degrades one strand of the DNA molecule and produces single-stranded DNA molecule. Transport of single-stranded DNA molecule across the plasma membrane is accomplished by the COM-E. COM-E forms the transmembrane channel. Finally, single-stranded DNA molecule gets integrated into the recipient genomic DNA by homologous recombination. Let us see the DNA uptake machinery in gram-positive bacterium Bacillus subtilis. COM-GC is the pseudo-palline complex. So this is responsible for DNA uptake. It acts as a piston by means of moving up and down in the peptidoglycan layer, and thereby it binds to the double-stranded DNA molecule and pulls the DNA molecule towards the plasma membrane. Double-stranded DNA molecule then binds to COM-EA. COM-EA is the DNA binding protein. Nuclease enzyme, that is N, degrades one strand of the DNA molecule and produces single-stranded DNA molecule. Transport of single-stranded DNA molecule across the plasma membrane is accomplished by COM-EC. COM-EC is the channel protein. Single-stranded DNA molecule binds with SSB protein, that is the single-stranded binding protein, for protection against degradation. Finally, single-stranded DNA molecule gets integrated into the recipient genomic DNA by means of homologous recombination catalyzed by brachioproteases. Artificial transformation. Natural competence and transformation are efficient only for the linear molecules such as the chromosomal DNA molecule. It won't suit for the circular plasmid molecules. So thus prokaryotic and eukaryotic cells need to be made competent artificially in the laboratory. Now we are going to discuss about the methods of artificial competence in bacteria. So this includes calcium chloride mediated transformation, freeze-thaw method and electroporation. Calcium chloride method. Calcium chloride method is the commonly used chemical method of making cells competent. In this method, bacterial cells are treated with calcium chloride. Calcium ion increases the permeability of cell membrane by means of making pores, which facilitates the DNA entry. This is the protocol for calcium chloride mediated transformation. Ampicillin sensitive E. coli cells are grown in LB agar medium. Ampicillin sensitive E. coli cells in log phase are then transferred to cold calcium chloride solution. Ampicillin resistant plasmids are then added to ampicillin sensitive E. coli cells which are placed in cold calcium chloride solution. Cells are then transferred to a water bath and heat shocked at 42 degrees Celsius. Some of the competent cells take up the ampicillin resistant plasmids and are transformed. The heat shocked cells are then spread on LB ampicillin agar and incubated at 37 degrees Celsius for 24 hours. Ampicillin kills the cells that lack the ampicillin resistant gene. Only colonies of E. coli that have been transformed by the ampicillin resistant gene will grow on LB ampicillin agar. Freeze-thaw method. Actively growing bacterial cells are pelleted down by centrifugation at 5000 rpm at room temperature. Pellets are washed, cells are then frozen in liquid nitrogen and stored at -70 degrees Celsius. Competent cells are thawed on ice and DNA is added. Cells are then incubated for recovery. Transformed cells are selected by antibiotic selection. Electroporation. Electroporation is a method of transformation. In this method, foreign DNA molecule is introduced into the host cell through the application of high voltage electric pulses. Bacterial cells are placed in buffer and put into electroporator. DNA is then added and subjected to high voltage electrical pulse of defined magnitude and length. The cells are then allowed to recover and finally selected by antibiotic selection technique.